In the field of biology, we are witnessing a quiet but profound revolution. Artificial intelligence is rapidly becoming an indispensable tool for deciphering the immense complexity of life. But this change is about more than just processing power. It's compelling us to ask a rather fundamental question. As we come to rely more and more on AI, are we also changing what it means to truly understand biology itself?
To really appreciate why AI represents such a significant shift, we first have to consider what makes biology so different from other sciences. In physics, for instance, we have elegant, often simple laws, like the law of gravity, that can predict the movement of planets with remarkable precision. But in biology, there's no single law of the cell that can predict everything it does. So why is life so much more complex, so much harder to describe with simple rules? That's the core challenge we're going to explore.
You can think of it like this. Physics often operates from the top down. It begins with a broad, general principle and uses that to explain specific observations. That's a deductive approach. Biology, on the other hand, almost has to work from the bottom up. We start with a vast amount of specific data from experiments and try to construct a broader understanding from all those details. This inductive approach isn't really a choice. It's a necessity for tackling the inherent complexity of living systems. Now, the traditional framework for scientific understanding is called the deductive nomological model. It's a formal way of saying that we understand something when we can show it's a logical outcome of a general law. For example, we understand why a spoon looks bent in a glass of water because we can explain it using the laws of light refraction. This top-down method has been incredibly effective in physics, but it relies on a key assumption that there are simple, human intelligible laws to be discovered in the first place. So what is it about biology that makes it so resistant to these kinds of simple laws? Well, there are three primary challenges. First, there's multidimensionality. A living system is complex at every single scale, from individual molecules all the way up to the entire organism. Second is conditionality. A cell's behavior can change dramatically based on its environment. A cell in a petri dish might act completely differently from one inside a living body. And third, emergence. You can know everything there is to know about the individual components of a system, but still be unable to predict how the whole system will behave. A protein's function, for instance, emerges from sequence in a way that's very difficult to predict from first principles. So, if the traditional tools of simple, universal laws don't quite work for biology, what's the alternative? This is precisely where AI enters the picture. It suggests that we might need a new way of thinking about what it even means to understand something so complex. Machine learning, with its bottom-up, data-driven methodology, provides us with a new set of conceptual tools. The goal isn't to replace scientists, but rather to offer them a new lens through which to view their data. The first of these new conceptual tools is the idea of understanding as information compression. You can think of it like creating a summary of a very long and detailed book. To do it well, you don't just pick sentences at random. You identify the key themes, the most important plot points, and use them to build a much shorter version that still captures the essence of the original story. In a similar way, AI can take massive biological datasets and find the essential patterns while filtering out the noise, effectively compressing the information into a more manageable and understandable form. The second tool is qualitative intelligibility. This is really about developing a strong intuition for how a system functions. You might not be able to write out the precise mathematical equations for a weather system, but you can look at a map of pressure systems and get a good sense of whether you'll need an umbrella. AI can help us build this kind of intuition for complex biological systems, often through powerful data visualizations. It allows us to ask what-if questions and get a feel for how the system might respond without needing to perform exhaustive calculations for every detail. And the third tool is dependency relation modeling. This is all about mapping the intricate web of connections within a system. It's like creating a detailed interaction map for all the molecules in a cell, showing not just what's there, but which components influence each other and how. A successful model of this kind isn't just a parts list. It's a map of the functional relationships that make the entire system work. All right, let's move from these abstract ideas to a concrete example of how they are applied in the real world. We're going to examine one of the foundational challenges in all of biology, predicting the three-dimensional shape of proteins. This is a problem that scientists have grappled with for decades, and it's an area where AI has delivered a truly significant breakthrough.
Here's the essential puzzle. Proteins begin as a simple, one-dimensional string of building blocks called amino acids. But in order to perform their functions, they have to fold into extraordinarily complex and specific three-dimensional shapes. The instructions for this intricate folding process are known to be encoded entirely within that one-dimensional sequence. The central question for over half a century has been, how can we reliably predict the final 3D structure just by reading that 1D sequence? For many years, researchers pursued two main strategies for this problem. The first was a mechanistic or physics-based approach, trying to simulate all the intricate physical forces that guide a protein into its final folded state. The second was an empirical or template-based approach. If you had a protein that was evolutionarily related to one whose shape was already known, you could use that known structure as a guide. What modern AI has managed to do is effectively integrate both of these ideas. This brings us to AlphaFold 2, the AI system that has produced such remarkable results in this area. Broadly speaking, it operates in three main steps. First, for its input, it doesn't just look at a single protein sequence in isolation. It examines a large collection of related sequences from different species to gain evolutionary context. This is known as a multiple sequence alignment, or MSA. Second, a core component of the AI, called the evoformer, processes all this data to infer which parts of the protein are likely to be physically close to one another. And finally, a structure module takes this learned information and uses it to construct the final 3D model of the protein. Now we can connect this directly back to our new frameworks of understanding. The attention mechanism within the AI, which determines which amino acids are most important for the structure, that is a form of information compression. The part of the model that calculates the distances and angles between different parts of the protein, that's dependency relation modeling. It's building that map of connections. And the system's ability to recognize a protein's shape regardless of its orientation in space, that's another sophisticated form of information compression using physical symmetries. So you see, AlphaFold 2 is not an inscrutable black box. It's employing these new forms of understanding in a very concrete manner. But this raises a critical question. Did AlphaFold 2 simply memorize a vast library of known protein shapes, or did it actually learn the underlying principles of how proteins fold? The evidence strongly suggests it has learned general principles. For one, it can accurately predict novel protein structures, like certain beta-selenoids, that are unlike anything in its training data. Also, when the AI reports that it's not confident about the structure of a particular region of a protein, that region often turns out to be intrinsically disordered, meaning it lacks a fixed shape in reality. In other words, the AI's uncertainty corresponds to a real biological phenomenon. There's even evidence it has learned information related not just to static shapes, but to how proteins move and flex. This leads to a rather compelling conclusion. Attempting to simulate protein folding purely from physics is computationally prohibitive. It's like trying to track the path of every single water molecule in a wave. AlphaFold 2 arrives at the correct final state with far less computation. So, one could argue that AlphaFold 2 provides a better reduced description of the folding process. It may not be a simple, elegant law that we can write on a blackboard, but it's a more efficient and practical model of the physical reality than what we had before. Let's now zoom out, from the level of a single protein to an entire community of cells. Our second case study brings us to the field of genomics, where a technology called single-cell RNA sequencing is creating another explosion of data. We can now measure the activity of thousands of genes across thousands of individual cells simultaneously. Here, the challenge isn't just acquiring the data, it's making sense of it. And for this task, AI is not just helpful, it is absolutely essential. The problem here is one of what scientists call high dimensionality. Imagine you want to plot a single cell on a chart. Instead of just an X and a Y axis, you would need perhaps 20,000 axes, one for each gene being measured. Now imagine trying to do that for 50,000 cells. You end up with a dataset so vast and complex that it's impossible for a human to visualize or find patterns in it directly. The role of AI, then, is to navigate this incredibly complex data space and reveal its hidden structure. So, how does an AI system make sense of all this? The analysis pipeline typically involves three key steps. The first is dimensionality reduction. The AI takes that 20,000 dimensional data and projects it down onto a 2D or 3D map that we can actually look at and interpret. The second is clustering. The AI examines this new map and groups similar cells together, much like identifying different neighborhoods in a city. And finally, trajectory inference. The AI attempts to connect these groups to map out a biological process, like how a stem cell might develop into a mature neuron over time. When it comes to creating these 2D maps of cells, there is a key trade-off. One approach, called PCA, is a bit like a standard world map. It preserves the large-scale distances and directions fairly well, but can distort local shapes. Another approach, such as UMAP, is more analogous to a subway map. It isn't geographically precise, but it excels at showing you which stations, or in this case, which cells, are close to each other and how they're connected. This UMAP method is a prime example of generating qualitative intelligibility. It provides scientists with an intuitive feel for their data. Both of these methods are, at their core, sophisticated forms of information compression.
Once this map has been generated, the scientific discovery process can begin. Clustering algorithms can then draw boundaries around the different neighborhoods of cells, which scientists can then investigate and label as distinct cell types. Trajectory inference algorithms can then draw a path through these neighborhoods, suggesting how a cell might travel from one state to another as it matures. These AI tools effectively transform a single, static snapshot of thousands of cells into a dynamic narrative of a biological process. So, from the intricate folding of a single protein to the complex society of thousands of cells, we've seen that AI is more than just a new tool. It represents a new way of conducting science. This isn't just an incremental improvement. It may well be the beginning of a new paradigm for scientific discovery, especially in a field as fundamentally complex as biology.
The reality is that in the 21st century, biology has become a data-driven science. We can generate more data in a single experiment than a biologist in the 20th century might have produced in an entire career. This has created a scenario where the bottom-up, data-intensive nature of machine learning is a perfect match for the bottom-up, data-rich nature of modern biology. So the essential takeaway is this. Machine learning is not just a tool for making predictions. It's a new engine for inductive reasoning. For biological systems, with their multidimensionality, their conditionality, their emergent properties, the traditional top-down, law-based approach often falls short. The bottom-up, pattern-finding approach of AI offers a powerful and necessary path forward. And this leaves us with a final and much broader question. We've seen that AI can begin to understand the world through frameworks like information compression and dependency mapping, which can be quite different from our own human intuition. As these systems become even more sophisticated, they might identify patterns and construct models that are highly accurate, but that we find difficult to comprehend. It forces us to wonder, as we continue to build these new forms of intelligence, what aspects of reality might they perceive that we cannot? And what might we be missing if we insist that the universe must be understood only on our own terms?